Hey guys, this review video is going to be about DNA structure and the replication of DNA. All right, so really quickly, let's take a look at the structure of DNA. DNA is an organic compound. If you remember organic compounds, Cody likes perfect nachos. It is a nucleic acid. Nucleic acids are made up of a monomer known as a nucleotide. So right here is a nucleotide. It has a phosphate, a sugar, and a nitrogen base. So if I take a bunch of nucleotides, so a bunch of monomers, and put them together, I create a polymer called a nucleic acid. So that puts the Na in DNA, nucleic acid. What about the D for DNA? The D in DNA comes from this sugar here. This is a special sugar called a deoxyribose sugar. So that puts the D in DNA, deoxyribonucleic acid. Now what I've created here is not DNA yet. DNA is not single-stranded, DNA is double-stranded. The two strands of DNA are going to bind to each other at the nitrogen bases in the middle. And they're going to use hydrogen bonds to do that. Let's kind of jump through this really quick. Here we go. So those nitrogen bases, there's 4 of them in DNA: adenine, thymine, cytosine, and guanine. Adenine always bonds with thymine, cytosine always bonds with guanine. We say ATCG to remember that. They're bonding with those hydrogen bonds, which are weak, so it takes multiple hydrogen bonds in between the strands. Now the backbone portion is called a sugar phosphate backbone. You can see it here: sugar, phosphate, sugar, phosphate, sugar, phosphate, and that keeps repeating. And finally, this structure is not flat. This structure is a double helix structure because it spirals on itself. All right, let's talk about replication. So here we have our original parent DNA molecule, okay? And we know we need to split it. So an enzyme comes in called helicase that is used to split it. So it splits it down the middle between those nitrogen bases. It's breaking those hydrogen bonds as it splits it. We then bring in DNA polymerase. So DNA polymerase is this guy. And you notice that there's one for each of the strands, right? So there's a parent strand here, there's a parent strand down here, and DNA polymerase comes in on each one. DNA polymerase, its function is actually in its name. It makes a new DNA polymer. So it's actually right here, it's making a DNA polymer, and it's doing that by bringing in nucleotides, which we know is the monomer, and it's pairing them up with the complementary bases, A's with T's, C's with G's, and we're making a brand new daughter DNA strand here, and another daughter DNA strand down here. So in the end, I have a daughter DNA molecule being created and a full daughter DNA molecule being created. So we're going to go from 1 to 2, and they're going to be exact duplicates and replicates of each other. Now this process is called semi-conservative, and here's what that means. The word semi means half, and conservative means to save. So basically what that means is we are saving half of the original, or half of the parent strand, and it's being used in each of the daughter strands. So here's the parent, and here's the parent. Now the reason why it has to be semi-conservative is so that it can reduce the number of errors that are taking place during replication. Cells want to have perfect replication every time. So they figure if they send a perfect copy to each one of the strands or each one of the molecules, it should make a perfect copy in the end. So it is semi-conservative to reduce mutations. Okay? Semi means half, conservative means to save. Half of the original is in each of the DNA molecules. Now, as I remember, guys, anything that ends with ACE, like DNA polymerase, helicase, ATP synthase, those are enzymes, and we know that an enzyme is a protein. All right, guys, so if you had any trouble with DNA structure, if you have any trouble with DNA replication, you want to make sure that you watch this video again or several times in order to really get these ideas across and build a solid foundation.